In this module, we will discuss another important branch of proteomics, and that is the comparative proteomics. That is where how we can compare the different proteins which are present in the different species. You see, proteins are important molecules, as we have said times and again, in a sense that proteins are those biomolecules which characterize different organisms and species. Organisms and the species, they are differentiated by their presence of different proteins. Now, a natural question arises that what makes a worm, a fly, a lion, a man. So we come to the answer that it is the proteins which are produced in these organisms that set them apart, that differentiate between them. And in order to study this further, in the beginning of this century, in 2009, Michael Hangeratner and colleagues, they examined the simple organism, that is C. elegans, which is a roundworm. Its proteome, its protein content, its protein significance, its protein sequence, and they use the quantitative techniques as we described in other different modules. And they discuss that what is the presence of the enzyme proteins which are in C. elegans, and they compared it to the Drosophila, that is the fly, the Drosophila melanogaster, its proteome, its amino acid sequence, which has been reported earlier in 2007, because a lot of work has been done on the fruit fly. Now, when they compared the data of the proteins which they obtained, they identified with the proteins which was predicted from the genome. The genome of these two species, these two organisms, they predicted the different presence of different protein. And they found that the certain classes of proteins which was present in the genome, that was underrepresented. That was underrepresented, not well represented in that data. So again, these scientists, they estimated the protein concentrations in C. elegans from their mass spectrometry with iCat data. You see, these are the techniques which are commonly used in the quantitative proteomics, and compared them with similar protein concentrations data from the previous Drosophila study, from the previous study of the fruit fly. They study their mass with the mass spectrometry, iCat techniques, and the other quantitative proteomics techniques. The protein concentrations of orthologs in worm and fly, they showed a much better correlation. What? The concentration of orthologs in worm and fly, they showed a much better correlation than which was observed, which was compared with messenger RNA concentration. So it means that the quantitative protein estimation, they showed us a very good correlation between the orthologs. Now, the benefit of the comparative study, what are the benefits of the comparative study? What are the benefits of the study or the observation or the conclusion which these scientists have drawn? That is, the mass spectrometry data can be used to compare protein concentration in two different organisms. That is understood that the quantitative proteomics can compare the different data of the protein concentration in different organisms. Now, this kind of analysis, when applied to C. elegans and Drosophila, it allowed that the concentration of orthologous proteins in the two organisms, they are correlated much better than the orthologous messenger RNA. So it showed us that these techniques, they give us much better result for comparing the data than was obtained with the messenger RNAs in these two organisms. And furthermore, they even did better than the proteins and corresponding messenger RNA in the same organism. So the quantitative proteomics, it helps us to determine a better correlation between the orthologous protein of the two organisms which we studied, that is C. elegans and Drosophila, and we can extend this result further to different species of organisms. So it can help us to determine, it can help us to study that what are the correlation between the orthologous amino acid, orthologous proteins, which will be presented in those species which are under study, and shall increase our understanding, a better understanding between the orthologs than we will get through the messenger RNA in the same organism.